Hi everyone, this is the very last lecture of organic chemistry. You made it, yay! Give yourselves a round of applause or a pat on the back. And we're actually going to just wrap up our chapter on amino acids, peptides, and proteins. So this will be a pretty short lecture, but what we'll really talk about is the structure of proteins and the different types of proteins that exist. But first, let's talk about a structural component of proteins, which is disulfide bonds. So, as you can probably guess, disulfide bonds occur between two sulfur atoms. And in particular, there's an amino acid that has a sulfur group or a thiol group in its side chain, and that is cysteine. So, if we draw cysteine, we can see that sulfur group on the very end, and I'm just going to abbreviate cysteine as R, and then I'll draw its side chain. But if we have two cysteine amino acids and we join them together, this would actually be considered an oxidation reaction. And that's because we're losing two hydrogens in the process. And then we end up with a sulfur-sulfur bond, which is a disulfide bond. Now this is really useful because this can help determine the shape of a polypeptide chain, which can then also eventually determine the shape of a protein. So for example, let's say we have a chain of amino acids, and I don't know, let's say we have alanine, then cysteine, then glycine, and then let's say we have a few others. What can happen is the peptide chain can kind of curl around so that eventually the side chains on two of the cysteine amino acids can meet up and form a disulfide bond. So once that bond forms, we've got something called a disulfide bridge. So there are a lot of peptides that exhibit this behavior. One of them is oxytocin, if you've heard of oxytocin before. So oxytocin is really important. It's a hormone that kind of regulates our emotions, and that does have disulfide bridges. Okay, so let's talk about protein structure. So there are 4 different types of structures that we can refer to for a protein. And remember, proteins are just extra long chains of amino acids. So the primary structure of a protein just refers to the basics. So this would refer to the sequence of amino acids that make up the protein. So we're not even really thinking about how the chain of amino acids is shaped or structured. We're just thinking about the basic sequence of amino acids. Then the secondary structure is where we start to think about the shape. So the secondary structure of a protein refers to the geometry or 3D conformation of certain regions of the protein. So I have a couple of examples for secondary structure off to the side. The first one on the top, this is showing amino acids in a chain that are kind of curling in on themselves. So they're forming something called an alpha helix. And what I'd like you to notice is that they're forming this alpha helix through hydrogen bonds between carbonyl groups, so that oxygen, and then a hydrogen from an amine. And these are just occurring all along this chain. So these are really just all the way down the chain, and this is what forms that alpha helix structure. There's also another structure that amino acid chains can form. One is a parallel beta sheet, another one is an anti-parallel beta sheet, and it just really depends on whether your C termini are lined up or if the N term or if they're opposite. So down below I have a parallel beta sheet where the C termini are aligned and the N termini are aligned. But you could also flip one of those and then it would become an anti-parallel beta sheet. So if one C termini was aligned with the other chain's N terminus. But regardless, it doesn't really matter in the long run. I'm just trying to show you that this is another structure that can form. And again, we see hydrogen bonding all along this chain here. So typically, when you see, for instance, a structure of a protein online, you're going to notice those alpha helices or beta sheets, which are usually shown as kind of a really thick arrow. And it just sort of shows the direction that the beta sheet is going in. And we'll actually see an example of this on the next slide. Okay, so the next type of structure that we can refer to is the tertiary structure of a protein. So the tertiary structure refers to the overall three-dimensional shape. So for example, in an aqueous solution, proteins will typically shape themselves so that polar groups are pointing out towards water and nonpolar groups are kind of inside, away from the aqueous solution. So myoglobin is shown off to the right here. This is a protein that stores oxygen. And notice these alpha helices that form in the structure. I don't see any beta sheets, but that's okay. So the polar groups, like let's say we have some R groups coming off here, those side chains. If they're polar, they're going to point outward. And then nonpolar groups are going to kind of arrange themselves inward so that they're not, interacting with water. Okay, and then the last type of structure is a quaternary structure. And a quaternary structure occurs when a protein has two or more subunits or polypeptide chains. So a really good example is hemoglobin, which is shown off to the right. Oh, I don't know what I was trying to spell there. Hemoglobin, there we go. So hemoglobin has 4 different subunits. You'll notice that they're in alternating colors. So 2 of the subunits are in blue and 2 of the subunits are in red. And all of these different subunits join together to create this much larger protein. And hemoglobin is a really important protein. It helps deliver oxygen where we need it. So there's also these what are called heme groups. And there's actually 4 of them, 1 for each subunit, and that's what holds on to the oxygen. So we again have 4 different types of structures. So the primary structure is just that basic sequence of amino acids. The secondary structure refers to the geometry at localized regions. So if you kind of zoomed in on your protein and just looked at a few amino acids in a row, you might see an alpha helix or you might see a beta sheet. And then the tertiary structure is kind of the bigger picture. So we're looking at the entire protein and its 3-dimensional shape. And then the quaternary structure is really only referred to if there's more than if there's 2 or more subunits in the protein. So for instance, myoglobin only has 1 unit. It's just itself, right? It's not really composed of multiple units there, so it wouldn't have a quaternary structure. But hemoglobin does consist of 4 subunits, so it does have a quaternary structure. All right, so now let's talk about different types of proteins that we might come across. The first type would be a structural protein. So a structural protein is used for structural rigidity, and these are typically fibrous proteins. So for instance, our hair is made of a polypeptide called alpha keratin. It's actually multiple polypeptides that kind of form an alpha helix. So we could write alpha keratin as an example. So that's what's in our hair, and that's what helps keep the structure of our hair. What else? Let's see. Then we have enzymes. And I'm sure you've heard about enzymes before, either in general chemistry or earlier in organic chemistry, or in your bio classes. So enzymes are biological molecules that help catalyze different processes. So for example, I always like to use alcohol dehydrogenase as my example. So if you don't have a lot of alcohol dehydrogenase, which is an enzyme, then you'll have trouble breaking down alcohol in your system. And that can cause you to feel kind of sick, or it can make you turn red. And then there's many other enzymes too. So there's lactase, which helps break down milk products. If you lack lactase, that can cause issues if you eat a lot of dairy products. What else? There's also amylase in your saliva that helps break down food as you're eating it. Yeah, there's a lot of different examples of enzymes that are really important in our bodies, and if they're not functioning properly, that can cause issues. Another type of protein is called a transport protein. So transport proteins, you can probably guess, help to transport molecules from one location to another. So we just saw hemoglobin on the last slide. That is a transport protein because it helps move oxygen through our system. All right. So that's pretty much it for this chapter. The sapling homework will be fairly short, but if you do have any questions on any of this content, just let me know. You can always email me or message me or set up an office hour using the Calendly link on Canvas. So yay! Celebrate being done with the lectures. You made it.